Hi, I'm Sriganesh Sharma. I'm a general surgery resident and a postdoctoral fellow from the University of Michigan. I just had the pleasure of presenting at Vascular Discovery 2022 on a panel that was talking about COVID-19 related thrombosis and therapies to address COVID-19 related coagulopathy. Our research at the University of Michigan focuses on epigenetic enzymes that modulate coronavirus-associated coagulopathy. We presented our research and our discovery about this particular epigenetic enzyme called MLL1 and how it regulates the production of procoagulant as well as profibrinolytic molecules after coronavirus infection. So briefly, we were able to do all of our work in cells and mice, and we're able to translate that into findings into the human disease. And our work lays down the groundwork and the rationale for targeting this particular epigenetic enzyme called MLL1 to address coronavirus-associated coagulopathy.
Hello everyone, I am Dr. Francois Briand, Director of Research and Business Development at Physiogenex. We are a preclinical contract research organization based in France, and we are using animal models of obesity and type 2 diabetes and related comorbidities like non-alcoholic steatohepatitis, diabetic nephropathy, and cardiovascular complication. We are using the golden Syrian hamster as a model for obesity and NASH. And actually, the golden Syrian hamster also serves as a model for SARS-CoV-2 infection, because the ACE2 of hamsters is really similar to the ACE2 of humans. So we actually wanted to use our obese NASH hamster model to see what was the impact on dyslipidemia and NASH. The main findings we found in this model after SARS-CoV-2 infection is that the dyslipid profile that we already have in the obese NASH hamster model is sustained or even more pronounced with elevation of free fatty acid levels or LDL cholesterol levels. And also we found that the liver lesions and liver fibrosis that we have in this model are also sustained and maintained during the SARS-CoV-2 infection. And this is happening despite a profound and substantial body weight loss due to the infection. So we would expect that with body weight loss, you would have actually some impact on liver lesions, but it is not the case. We still keep these advanced liver lesions and fibrosis. So we are still trying to understand the mechanism by which we have this pro-inflammatory and pro-fibrotic profile in the liver, as well as the more pronounced dyslipidemic state. But we think that this hamster model will be really useful to understand the severe forms of COVID-19 and eventually evaluate some therapies that would help to treat patients that develop the most severe forms of COVID-19.
Hi, my name is Connie Hess. I'm an interventional cardiologist at the University of Colorado. I had the honor of presenting the Aspen COVID-19 data here today on behalf of the Executive Committee and investigators. So we know that patients with COVID are at risk for thrombotic events despite use of traditional anticoagulants. And so we know both the processes of inflammation and thrombosis are at play here, and that tissue factor is an important contributor to both. So what we were studying in Aspen was the use of recombinant nematode anticoagulant protein C2, or RNAP-C2, which is a novel tissue factor inhibitor that also has anti-inflammatory and antiviral properties. In Aspen, RNAP-C2 was compared to standard of care heparin, 2 different dosing regimens, and we found that the drug was safe, that there was no significant increase in bleeding or adverse events with the drug. The primary efficacy outcome was change in D-dimer, and there was no statistically significant difference between RNAP versus heparin, although we did observe some interesting trends with greater reductions in D-dimer with RNAP-C2, though not statistically significant. So I think these are very promising data that support the safety of looking at RNAP-C2 and not only in COVID, but also in other disease states where tissue factor may be driving adverse events.
And those are some of the highlights of the session for today at Vascular Discovery 2022.